Thanks so much for having me on. I'm really excited to be here. So I want to tell you a little bit about my work in language models. And so I'm going to go kind of in order of, I would say, machine learning, sort of deep learning, which is where we train models specific to our applications. Then I'm going to show you increasingly how training models and designing architectures has become less and less important, and how we're reaching this kind of transition point and thinking about how to do tasks like drug discovery. And I have an opinion article coming out in Nature Reviews Chemistry soon that just lays out some of these ideas. But of course, I'm happy to share it all with you today. So I want to start by visiting sort of an older paper now. This is maybe 6 months old work. And it was about this idea that we've gotten really good with these pre-trained models. So a pre-trained model is something like ESM, which is from the Facebook group, or something like AlphaFold you can kind of think about as a pre-trained model, and start using these things for forward design. Right, so we've gotten really good at these machine learning tasks where we can look back and make sure we can predict what happened in the past, but we want to start predicting which sequence to try next, which drug to try next. So I want to talk a little bit about this forward-looking process. And this is kind of called like learning with a few data points or experiment design. And I want to break down just some nomenclature here to begin with. So active learning is basically when we want to build a very good model. So we're going out and we're gathering data, and we want to use that data to predict what is a really good machine learning model, like predicting affinity of a drug, predicting if a sequence will bind to a protein, things like that. Then Bayesian optimization is if we just want to get a really good structure, we want a really good sequence. So now we're not so concerned about building a good model, but just getting the best point. And then the last kind of setting is something called a bandit problem. And there we're trying to find sort of a series of steps that lead to each one of them being good. So maybe if you're doing drug discovery and you're trying to optimize one property, but you want to keep another property constant or still good, you might try something like a bandit problem as you go through the steps. All right. So I'm going to just do a little bit of math here about Bayesian optimization. So in Bayesian optimization, we have, in this setting, we have X being a sequence. So this could be a molecule, it could be a peptide. And then in this project, I'll be talking about as a peptide. F of X is our experiment. So this is like an SPR affinity measurement, or this could be an animal model or something. And then Y is the outcome of that experiment. So this is what we measure. And then what we're going to be using is an object called a surrogate model. And a surrogate model is our prediction of the outcome of that experiment. And in Bayesian optimization, so this is the goal of Bayesian optimization, is to find the best sequence or to find the best molecule. And you can see this setting maps, really well to something like drug discovery, where the surrogate model might be something like a biophysics or QSAR or something like this. Now, in Bayesian optimization, a lot of times people like to visualize it like this. We have a red line, which is showing us the sort of true f of x. Like if we had perfect information, we could visualize every single x value, every single molecule, every single possible sequence, and we could see their outcomes. But of course, we only see some of that. So the blue ones are the experiments we did, and the orange line is our guess at what the red one looks like. And you can see how we update our estimate of what the red line looks like as we gather more and more data. And the point of Bayesian optimization is to just find that maximum, that highest point on the red line. All right. So that's Bayesian optimization. So pre-trained models, these are these really exciting things like GPT-4, GPT-3, these ESM models like this, or there's some work from NVIDIA, these BioNema has a lot of these pre-trained models. And these things are really good at accomplishing tasks with few data points. And so in this project, we wanted to know if we could combine pre-trained models, which are very good at doing tasks with very few data points, and using them to do Bayesian optimization. Okay, so basically, pre-trained models we know are good at doing work with a few examples, but can we do that to then choose which thing to try next? Okay, there's a few problems with doing this. And so I'm just going to talk, just go quickly over these things. But to do Bayesian optimization, you have to have a measure of confidence. That's the difference between something like, just greedy methods or just always choosing the best compound, right? Versus trying to maximize exploration and exploitation. That is, sometimes you want to do an experiment not because you think it's going to be the best compound or the best sequence. You want to do an experiment because you want to learn something. And so this is kind of this philosophy of each time you do an experiment, you imagine you're buying information rather than buying, the best compound or the best sequence. And another challenge for this, so not just having uncertainty, so we can measure that confidence, that uncertainty, which gives us that trade-off between exploring and exploiting. We also need to have accurate uncertainty at low data. So calibration is another word for accuracy, but it's accuracy of uncertainties. So accuracy is like how correct you are, and calibration is how correct your uncertainty estimate is. So historically, the approach to doing this was something called Bayesian neural networks. So if you have neural networks and you want to do Bayesian optimization with them, you need to use Bayesian neural networks. I'm going to just sort of skip over this part here, but there's been some work that's shown you can actually approximate Bayesian neural networks by just using an ensemble of neural networks. So it's not so critical here, but basically the idea is that what we want to do is, instead of taking a pre-trained model and then making all of the trillion parameters in it, well, not quite a trillion, but hundreds of billions of parameters, we want to make them all themselves uncertain. We can actually only make the top of the neural network, just at the very end of the neural network, have uncertainty, and that will give us uncertainty in our predictions. And so in this project, we built a model that looks like this, and I'll just kind of walk you through it. Is that what we do is we start with a collection of sequences and labels. So we have peptides where we've measured the properties of them. Then what we do is we take a pre-trained model, in this case, UniRep. So then what we do is we train an ensemble of the top of them, the head of these neural networks. Okay, so we have a pre-trained model that's seen already, a billion sequences or hundreds of millions of sequences, and we're just doing a little bit of extra fine-tuning on the top of that to give it this uncertainty. Then we have a model which can predict the outcome of an experiment with uncertainty. Then we do this Bayesian optimization where we go and we try to find which new sequence, which new peptide can we try that balances exploration and exploitation. And that peptide is represented with a probability distribution. This is just sort of an implementation detail to get this kind of global optimization. And then we get a new sequence out. So then after doing this process, it says, go and test this new sequence. We test that sequence, and then we have a new experiment, and then we can repeat this whole process. All right. So the first thing to check is, does this kind of a model give us uncertainty? So to do this, we need to actually come up with tasks to try. So the difference between a lot of previous work in this setting is that we're doing something prospectively. We can propose any sequence. We can propose any molecule. So we can't just work on historic data. So we came up with a few increasingly difficult tasks. One of them is called, guess the sequence. This is Wordle. I don't know if any of you guys were around back in the day, about 12 months ago, everyone played Wordle. But so this is like there's some unknown sequence, and we can only try to see how close we are to that sequence and get feedback. This is just sort of a simple task that is nice because you can try it cheaply. Another sort of example is we can take a previously trained model, so like a simulation or an old machine learning model, and pretend like that's our experiment. So then we're going to try to maximize that previously trained model. And then the last one I'll talk about today, of course, the one we should be doing is doing what laboratory experiments, but for the purposes of this paper, it's we want to try a lot of different ideas. So we wanted things that are computational so that we can try them out. The last one is we use AlphaFold to do a peptide protein docking as our experiment. Now, I want to mention here, there's a lot of great work on using AlphaFold or the Rosetta Fold methods to do binding prediction, like to find binders. Those methods use derivatives. What we're doing here is we're treating this as a black box program, so we're not seeing derivatives. So I'll show you these results in a moment, but just understand that this is not the best you can do. We're kind of artificially handicapping ourselves by viewing AlphaFold or Rosetta Fold as a black box model. Now, what's nice about that is that if you ever want to do something where you maybe do Rosetta Fold, then you do a minimization, then you do some other tasks, those things break your gradients. So you can't do like RF diffusion with molecular dynamics at the same time. So this kind of work I'm talking about is really nice because it could be anything. It doesn't matter what it is. Whereas if you're specifically trying to do AlphaFold peptide protein docking, there are some better techniques than ours. All right, great. So I'm not going to go into the results too much, but basically one of the things we want to make sure we check is we want correlation to increase as we look at more data, but we also want log likelihood to increase. Log likelihood is that measure of calibration. So making sure that you are both accurate and you have correct uncertainties. Let's go ahead and see some examples. So the first example I'll show you is predicting if a peptide is hemolytic. So we want to predict if a peptide is hemolytic for, sometimes it's a goal for what we're working on, or sometimes it's something we want our peptides to not do. And so what I have on the x-axis is number of peptides we've tried. So if we just randomly try a peptide sequence, we're seeing this score, and the score here is like a probability of it being hemolytic. So this is just a mapping. So like the actual test for hemolysis is like a dose response experiment. So we're just mapping that to some 0.1 measurement. So if you just try a random sequence, you get an average of about 10% of the time it really is hemolytic. So what we do is we repeat this, kind of pretending like we're doing these experiments. We repeat this over and over again. So we have no data, then we get one data point, and then we propose a new sequence. Then we get 2 data points and we propose a new sequence. Then we go up to 10, to 20, to 30, to 40. And this red line is kind of like the most hemolytic sequence that's ever been found. And you can see after, say, 20 experiments, we have a very hemolytic peptide. So something that's just quite hemolytic. So this is nice. We can see, knowing no information about hemolysis after 10 experiments or so, we're pretty close. After 20, we have something quite hemolytic. But now let's get to something a little bit more authentic. So, sorry, this is still working. Yeah, great. So this one is something called RAS GTPase. This is a like KRAS. This is an interesting drug target in cancer research. And what we've done here is we've basically cut away its binding partner, the SOS protein, and we want to find a peptide which binds in the same spot. So that cut away, that helix right there, that's the SOS cut down to where it binds. And then what we're going to do is do this forward experiments Bayesian optimization to try to find a binding peptide. So each red one here is a new sequence being tested by the algorithm. So it's proposing sequences, their variable length, and it's doing an AlphaFold calculation and seeing how strong the binding is. So the way we assess binding here is we look at 2 different measurements. One of them is the PLDTT of the peptide. So that is like how confident it is in the bind, and the distance is how close it is to the site we want. So we start with, no information, then we do one experiment, and basically over time, as we try more and more peptides, we get to a very, we kind of lock in the distance pretty quick, and then we get a sort of a rise in the PLDTT. Now, what's nice about this approach is that we're not relying on derivatives, like I said, so we can do things like use different kinds of, like the multimer model of AlphaFold, which doesn't do well with propagating gradients. And we can also do a lot of recycles, which can help predict the bind, but it also shatters your gradients. So I would say there are better methods, like arc diffusion is probably a better method if it works. There can be problems with that approach. But this is a nice black box method, so it works on any kind of protocol you can come up with. All right, so I've shown you that basically using large language models, like pre-trained models, we can do this kind of design of sequences with only a few data points, and we can do it with calibration. So we have confidence measurements, and we can do this, as a design task. So proposing which peptide to try, it can do it in batches as well if you want to do like 30 peptides at a time. There's some other work in the paper I didn't show it here, but this does better than if you just start from scratch. Then a lot of the work that we do is kind of open science. So I'll just show you quickly what is the interface for this. So you basically have a couple lines to import this code, and then you tell it a sequence, and then you tell it the affinity or the score, whatever you're trying to optimize. Then you tell it another data point, then you can ask it, and it will tell you which one to try next. And then you can ask it what would be the best one, and you could, of course, make predictions. So it's a really nice, simple method to use to do any kind of sequence optimization task. So it can be peptides, it could be proteins. All right, great. So that was kind of like, I would say, more traditional machine learning. And then in August of last year, I started doing red teaming for OpenAI. So red teaming meaning I was one of the people who had access to GPT-4 very early. I had it about 6 months before everybody else. And my goal there was to sort of see like what's possible with it, and more importantly, what ethical or or misuse could be done with it. And I really noticed that GPT-4 was quite remarkable in a lot of different tasks. And this shouldn't be a surprise. So language has been sort of civilization's greatest collaborative project. We've been working on language as a tool to go across biology and chemistry, material science, and everyday life. So language is something that really has been honed to work well across all these tasks. So it shouldn't be that surprising that large language models like GPT-4 can work well on both reading a paper and also looking, at the newspaper. And so since my work, I've had a lot of papers and we've done a lot of interesting ideas, and sort of the North Star, sort of the ideas that have been guiding our work lately is that one, that language is a fundamental representation of science. So that is, in chemistry, we have this idea that the chemical structure kind of unites all of chemistry. But I think, more broadly across many different fields, language is a really powerful representation that can connect things that maybe are not so obvious. If you've ever tried to work with something like NIH databases or the European equivalent databases, you have these IDs, you have paper titles, you have the names of proteins, you have gene names, etc. But you'll find out nowadays, if you use GPT-4, it actually has a shared embedding of all these different names and titles. And so language, to me, I think is a more robust kind of universal identifier for a lot of the concepts in biology. And then number two, I'll talk just briefly about is that deep learning models should be able to work in language as well. So that project I just showed you, it outputs numbers, it outputs predictions, but I think there's been some really great work on making deep learning language models or deep learning models from any domain work with language. And then the last one is that I don't think that large language models are going to replace all models. I think there's still, obviously there's still room for biophysical models and for classical machine learning models. But what large language models I think will do is they will be the glue or the sort of agent that connects all the different models that we can use in biology. All right, so let me start with getting basically, how do we get models to explain themselves? Okay, so we're really good at predicting things like if a molecule will cross the blood-brain barrier. This is kind of surprising because I think biologically this is a challenging problem. But if you go and you try to train a machine learning classifier in predicting if a molecule will cross the blood-brain barrier, you might be surprised that you can get accuracies of about 95% or so. And one of the questions that arises is why are they so good? What are they learning? So one of the things we can do is we can get deep learning models to actually output language explaining what they're doing. And so let me just give a little bit of explanation on what this means. So a prediction is like when a language model predicts an outcomes, or it doesn't have to be language. When a machine learning model predicts an outcome, like if a molecule will cross the blood-brain barrier. So when we make a prediction like this from any method, there's a justification. A justification is like why you should trust a prediction. For example, if I predict that this peptide is going to bind to the protein, a justification might be like my model is 90% accurate. And then interpretability is like if you can look at the model and reason about why it makes predictions. So, for example, if you have a molecular dynamics model that predicts free energy, you can go and watch the simulation movie and kind of look at why the free energy is predicted to be the way it is. Or maybe you have, a QSAR model and you can just look to see what descriptors are important for it. Then an explanation is an extra piece of information for humans. And this is supposed to give you a little bit of, it's supposed to give you context and cause for why the prediction was made. And one way that we can do this is counterfactuals. So counterfactuals is like, my grant proposal was rejected from NSF. This is a true story. If I correctly formatted my references, it would have been funded. So this is the counterfactual, which is a very similar setting, but a different outcome. And it's an explanation for the original outcome. So the rejection can be explained by the counterfactual. And in chemistry, what does this mean? In chemistry, it's like if I have a molecule here on the left, which is not supposed to cross the blood-brain barrier, and then I have one on the right, which is supposed to cross the blood-brain barrier. This one is very similar to the other. It's like counterfactual, and it tells us a little bit about why this is not crossing the blood-brain barrier. All right. So again, I'm going to skip over a little bit of the mathematics here, but the idea is how do we construct these counterfactuals generally? One way to do this is to explore molecular space. So I have a molecule on the top left, and I have an outcome, and I want to explain the outcome. So what I can do is enumerate lots and lots of similar molecules, similar compounds. And the little highlighting here is just showing the change I made to the molecule. And so what we do in this setting is that we have a molecule we want to predict, and I've represented all the molecules as dots here. And then what I can do is find a similar molecule, which I've highlighted here in yellow, that's very close to it. So these little dots are all compounds, and how close they are in space is showing how similar they are. And this one that's very close to it, that's yellow, it has a different outcome. And so this is kind of like, the counterfactual. So I have a molecule which crosses the blood-brain barrier. If I delete the carboxylic acid group, it will not cross the blood-brain barrier. So I think that's a really great method. And there's a kind of an analogy for medicinal chemists called match molecular pairs. But we're not quite at language, right? So the goal of this work is to have these models not just explain using something like a counterfactual, but explain in English. So we go a little bit farther now, and instead of just looking at these dots and only finding the closest one, what we can do instead is fit our own model to those dots. So we want to predict this base molecule again, and then we have all these little dots or compounds that are changes to it. And now what we're going to do is we're going to take our deep learning model. So remember, we were predicting if a molecule is crossing the blood-brain barrier. Now we're going to predict if a molecule is soluble. Why are we doing solubility now? Because I think most people with a little bit of organic chemistry background know what the explanation is for solubility. So we're going to change the example to solubility here. So I have all these little dots here, and I have a very good solubility predictor in black. And then what I'm going to do is around the molecule I want to explain, I'm going to fit a surrogate model. And this surrogate model is going to be really simple. It's just a linear regression. It's a linear regression, though, over human concepts. So what we do is we make a big warehouse of human concepts about chemistry, like number of acid groups, how hydrophobic it is, what are the functional groups present in it. Then what we do is we fit all of these human interpretable concepts locally on this space of molecules. Okay, so each time we want to explain something, each time we want to explain one molecule, we fit a brand new linear regression model around that using our solubility predictor. And I'm showing you here, like we fit it in this example to functional groups, then we can show which functional groups contributed to the solubility. If you know a lot about organic chemistry, maybe you can spot, on the heat map here that yes, the parts of the molecule that are important for solubility are highlighted. Okay. Again, in the interest of time, I think there's lots of exciting stuff here, so I'm not going to get into too much details on the implementation, but essentially what we're doing is a weighted least squares to fit our human concepts or human interpretable concepts. And then we just do weighted least squares because we want to have a lot of weight on the molecules that are nearby our interesting molecule. So here's some examples from this method. So I have a molecule I'm trying to predict solubility. Then on the right side, I'm showing you which human interpretable concepts or descriptors are important. So the bar is showing the length of the bar is how important it is, and the direction is whether it makes the solubility go up or down. So it turns out that hydrophobicity is the most important predictor for solubility, which is very reasonable. For this specific molecule, remember, these are explanations for this specific molecule. The number of aromatic bond groups is important. The number of acid groups is important. The number of hydrobond donor acceptors is important, and the basic group count is important. One of the cool things about this method, too, is that these bars are actually t-statistics. So we can use a p-value or a t-test to assess which descriptors are really important and which ones are kind of coincidental. Then we can map it to something called max descriptors. Max descriptors are yes/no questions that a medicinal chemist might have about a molecule. So things like, is there a heteroatom in a six-membered ring? Is there an alkene bond? Questions like that. And then we can, of course, map it back to functional groups as well. Now, What we have done now is we've taken a deep learning model. It could be any model. In this case, it was a deep learning solubility model, and we've mapped human concepts as the explanation. And then what we can do is take all of these human concepts that were important and feed them into a language model. And then the language model can give back. So all we're doing is we're just taking all these concepts that we now know are important, and then we feed them to GPT-4, for example, or ChatGPT. And it summarizes this into an explanation. So the presence of a carboxylic acid group and this hetero-N basic group on the molecule are positively correlated with its aqueous solubility. The carboxylic acid group is especially important as it is highly polar and can form hydrogen bonds with water molecules, allowing the molecule to dissolve in water. And so basically we can get out now a paragraph of text. So I don't have the slide on this, but basically what I've shown you is now we can go from data, we can fit a deep learning model, and then we can have the deep learning model give out predictions with explanations that are a paragraph of text. So any kind of setting where you're predicting the outcome with machine learning, this even works with black box models that are like biophysics models. Now you can have these models give back a prediction with an attached paragraph of text explaining why it made the decision it did. Okay, so I think what I've showed you now is machine learning with pre-trained language models, and now we've seen that we can get these models to explain themselves in language. But now let's go even farther down this idea of language. So I'm going to now move into something which I would say is much more speculative or much more controversial. Sorry, my wife got me an iPad for Christmas, and so I'm still learning to draw on the iPad. So please pardon my my penmanship here. Okay, so here's an interesting argument. Okay, so when we make molecules or we make anything, we have this idea that we synthesize it. Okay, the synthesis we all agree on, right? Like that's something that's literally how we make it. There's no ambiguity in synthesis. Of course, multiple synthesis procedures can lead to the same molecule. People cannot be clear on how they synthesize things, but that's literally what we do to make a compound. Then we have the molecular structure. And if any of you have taken quantum chemistry, you'll know that like a chemical bond is something that doesn't really have a precise definition. There is no Hamiltonian, or there's no quantum mechanical operator for chemical structure. So this is just like a mental model. We all believe in chemical structures, and I think medicinal chemistry, it's a very safe bet, right? But it's just sort of a mental model of what we've actually made. And then we measure the properties, right? And these are the real measurables that matter. And so I'm going to show you a little bit of an argument about like why the structure is latent. That is, we don't need this mental model of structure. We can actually work from synthesis directly to properties. And it saves us something that's really important in these molecule prediction tasks, is that if I propose a synthesis, it's obviously got a synthesis, right? But if I propose a molecule, it may not have a synthesis. And so there's some benefits to just forgetting about molecular structure and just working in synthesis and then predicting properties. Okay, so let's take a look at, here's just two little examples here. So this is kind of a famous example on top. So we have thalidomide, which is a molecule with two stereoisomers. In one configuration, it causes birth defects, and in one, it's an anti-nausea medication. So if you think about molecular structure, the Tanimoto distance between these two is basically zero. It's very similar, the same similarity, but very different properties, right? So this is an example where we have, almost identical structure, but very different properties. And down here we have two molecules that are very similar structurally. We just have, a little bit of a rotation of this group. It's a different connection there, different bond. But one of them has like a 30-step synthesis route, and one of them has like a 2-step synthesis route because there's a natural product available. And so these are two very similar structures, but very different synthesis procedures. So, this is just little anecdotes, but there are examples of why molecular structure is really not it doesn't have the benefits we think it has a lot of the time. Even though we may have two very similar structures, we can have very different properties or very different syntheses. So if we don't have to use molecular structure as a representation, there are advantages of skipping over it. So I want to talk now about using large language models to predict properties. So we know large language models are really good for maybe you're writing a poem, or large language models can maybe answer a question for you. But there's been this work back in December or so that showed you could actually take a table of data, in numerical data, and if you put it into complete sentences, then you can actually use it to do classification. So we have a famous data set example here, and we're taking basically the text, right? So this is like machine learning. So we have data, and then we're turning it into sentences. Then the language model completes the sentence, and then that sentence completion is our classification. All right, so pretty straightforward idea, but remarkable. It works quite well. And what's really powerful about this idea is you don't actually need to take the time to build a machine learning model. You don't need to go learn, what's a good architecture, what are the good hyperparameters, what are the features. You can just throw it all together into a sentence, and it works pretty well. So it's a great paper by Kevin Jablonka and Baron Smith and Philippe Faller. And what they did here is they showed if you just take GPT-3 and you do this lift, this fine-tuning procedure, you can predict a lot of properties like solubility, color. And here, this is working with materials. You can predict the phase of a high entropy alloy. You can predict lots of properties better than specialized deep learning models by just taking a language model. So just taking GPT-3, putting in an English sentence describing the material, and you can predict the property better than all these specialized models. Now, what's really powerful about this idea is that it makes the structure, it makes what you've made or what the actual compound is, it makes it all latent. And what's so funny about this paper too is that they used, so predicting molecular properties, they found that the IUPAC name works the best. So no smiles, no selfies, no inchi keys. The molecular structure to predict a molecular property, just the IUPAC name of the molecule is what works well, which kind of upends a lot of work on graph neural networks and, how do we embed molecules? How do we featureize molecules? How do we know what are the right descriptors? Just the name of the molecule. Pretty interesting result. And now let me show you how we go even farther, right? So in the beginning, I said Bayesian optimization is really powerful because it allows us to do design of experiments. It tells us which experiment to do next. So what we did here now is we took the idea from Kevin's paper, and now we took the idea of synthesis being what's important. And what we're doing is we're taking a paragraph of text describing how we make something. In this case, we're doing catalysis. Okay, so we have a sentence about how we made the catalyst. We don't even think about what we're making. We're making a zeolite here. I'll just tell you guys we're making a zeolite, but we didn't tell the language model we're making a zeolite. We just tell it how we mix things, how long we baked it for. Then we said what the reaction conditions are, what the flow rates are, and then we ask it to predict the yield. And we give it as a multiple choice question. And we give it as a multiple choice question because through some tricks, we can turn a multiple choice question into a probability distribution. And the reason why is that when you say, okay, what's the right answer, A, B, C, D, E, or F, the language model will give you back basically a score for A, B, C, D, or F. That is its probability of each of these letters, and that you can construct a probability distribution from it. Okay, so here we've dropped molecular structure. We don't care what we've made. We're just putting in English sentences about how we set up our experiment, and we're asking it to predict the outcome. And we're trying to do it with uncertainty. Okay, so let's go back one step to make sure this works with the solubilities, with the names. So here we have the solubility of 1-bromopropane is minus 1.73 log S. So this is, I'm predicting the solubility of a compound by just putting its name in. And what I'm showing you down here is that we can get uncertainty from this. Okay, so we get not just the solubility prediction, but the solubility prediction with confidence intervals. And these are a few different large language models, DaVinci, GPT-4, fine-tuning. So this GPT-4, we don't even fine-tune it. We just literally give it a few examples of other compounds that we know solubility for, and then ask it to predict the new one. And Gaussian process regression is just a baseline kind of traditional model. So GPT-4 predicting the compound solubility quite well. Now, I've kind of just skipped over this, but you have to give it a few example solubilities. Like, it just won't do this blind. You have to give it some examples of known solubilities. But what's really incredible about this procedure is that it just works with, English. There's no training. There's no, careful tuning of the model. You just give it a few examples. I won't talk about this here, but this is just showing how good this model is against other kind of models. All right, so then now, here's the example where we're really just working with a description of how we made the catalyst, or how we made the material. And basically with GPT-4, we're getting, good correlation, good uncertainties. All 6 of these are with large language models, and we're getting basically state-of-the-art results working with large language models. And then finally, the goal of all of this is to do Bayesian optimization. So here, the first thing I want to check is that as we add more data points, that is, if I propose this experiment, I propose this catalyst, I test the catalyst, and then I can go see how well we did, we improve, right? So like, as I do more experiments, the accuracy goes up, so we can still do learning here within context learning, and then we can do Bayesian optimization. And now, I know some of you are thinking like, oh, this is catalysis. What does it matter for me? I don't do catalysis. But remember, it's in English. So as long as your experiments are in English, you can describe your experiments in English, and you can, put it into a paragraph of text. This method will work. And in the paper, we did it for solubility. We did it for catalysis. And certainly we're working on doing it for things like sequences and compounds. But so here, what we're doing is now we're doing this forward task. So can we not just predict the outcome, but can we do design of experiments? So we start with no information, and then we do one experiment, and then we see what is the yield of the catalyst we got. Then we do 2 experiments, et cetera, et cetera. And so we can get to yields that are very, very high yields after, say, 80 experiments. And just for record, so we're getting to 20% yield. I think the thermodynamic maximum here is like 28%, and the best reported literature is 21%. So after 80 experiments trying catalysts, we can get up to pretty good yields. And by the way, an experiment here is like changing the reaction conditions, changing the synthesis procedure. Obviously, making 80 catalysts is a lot. It's a lot of work, but it's pretty cool that we can guide the design of these catalysts without ever stopping to characterize them, right? A lot of the work in catalysis research is that you need to know what you made so that you can understand the process. Here, we're just working in English. So imagine doing chemistry where you don't stop to see what compound you made. You just try different reactions. It's insane. So this code you can try out as well. You just tell it in English what you did. So 1 bromopropane, then the measurement, and then you can ask it to predict, and then you can ask it to choose which experiment to do next. And this, the English description here is compound names in this example, but of course we did these paragraphs of text. So you could try it out on your setting next. All right, great. So that's all really cool stuff. I think it's really exciting how far we can push it, but now let's go one level higher and start trying to automate, the whole drug discovery process. So there's this idea of neurosymbolic computing, or agents as they're more commonly called now. And the idea is that you basically take these large language models that can do these tasks, and you're going to give them tools, basically. If you take GPT-4, for example, and you ask it to multiply 2 very large numbers, it won't always get it correct. But if you say GPT-4 here's a calculator, and then you have it multiply large numbers, and you tell it to use the calculator, it will learn how to use the calculator. These things know how to use tools from English descriptions of the tools. So let's start with a simple example. Okay, so I have here ChatGPT, or GPT 3.5, and I ask it, what is the reason for making a bispecific antibody for CD38 binding? So it gives me back one of these sort of, ChatGPT answers where it talks a lot about the setting, but it misses the bispecific part, right? It gives a lot of information about like a monoclonal antibody, but it doesn't really give me something, what I need for the bispecific part. Now, instead, I start giving it tools. And now the tool I'm going to give it is basically access to scientific research papers. I won't talk too much about it, but we have this thing called Paper QA that basically goes and you ask a question, and it goes and it searches all of scientific literature, pulls up full text papers, cuts the papers up into relevant sections, and then gives it to these agents so that they can answer questions with context from all of scientific literature. And then you go back and you ask the same question, and now it gets it right that it's from multiple myeloma, and it understands what the bispecific antibodies are for and what their binding partners are, and it even tells you what paper it came from. So this tool, PaperQA, I think it's a really powerful tool to do literature search now, because it gives you in-text citations. It pulls from all of scientific literature, whereas ChatGPT just has memorized some fraction of scientific literature and can't cite its sources. So PaperQA kind of gets around this hallucination problem that we all know is an issue when you try to use GPT-4 or ChatGPT for doing scientific literature review. Okay, so that's one tool. So I just showed you that we can give this PaperQA thing, it gives these large language models the ability to know all of scientific literature, basically. Then we can give it a web search so that it can access anything, on Google. Then you give it a calculator, you can give it Python, you can give it a synthesis planner so that it can actually propose synthesis. So these models are not very good at basically planning out a chemical synthesis. And you give it access to write its own code. So this is the example that we did in GPT-4, was design a compound with the same mechanism as dasatinib and purchase it. And so we gave it all these tools, tell it to use the tools and get to work. Some of the tools in here is a patent search as well. I didn't cover all the tools, just patent search. There's also like if it's purchasable from a contract research organization. Okay. So it breaks this down in this question, thought, action, and observation. And so I've told it to find a drug that's similar to dasatinib. So then it wants to find compounds that have the same MOA as dasatinib. So then it goes and does a literature search for this, and it finds a few research papers that describe other compounds that have the same MOA as dasatinib and the same target. Then it decides it wants to take some of these compounds and modify them. So it gets the compound names, then it uses a tool which turns a compound name into the compound structure, here represented as smiles, and then it wants to modify this to make it novel. We have another tool, it's kind of like a patent buster. It's not very good, it's just sort of a proof of concept that basically modifies the chemical structure until it gets out of, until it gets out of the patent space. So then it comes up with a compound which is not patented. Checks to make sure it's novel, checks to see if it's purchasable, and then it purchases the compound. Okay, and then here it now has the final compound, and it purchased it from zinc, basically. It found it in enamine real. Okay, so that was sort of a simple example. Okay, now we've gone farther, and now we have safety assessment, we have getting the price of compounds, we have reaction prediction, reaction classification, we have a better sort of modification method, so we can propose lots of similar molecules in a more reasoned approach. And the way this works now is like, here's a slightly more sophisticated example. Synthesize a molecule similar to paracetamol that doesn't contain a methylamide group, then find the price of this molecule if it's above $500, plan a synthetic route. And then it goes through sort of a complex workflow. We call this tool ChemCro, by the way. So you basically get the smiles of the compound, then it finds the functional group in here, then it modifies it to remove that group, then it gets the price of this changed group, and then it decides that it's expensive, so then it proposes a synthesis route. And with these tools, it does quite a good job, actually, and we have organic chemists check this to make sure that it's reasonable. I won't talk too much in the interest of time about how we assess these things, but sort of the ultimate assessment is that we have it hooked up to a robot, ChemSpeed, and so it actually plans out its own synthetic route. We have an example that should be done today, I think. We've already executed the reaction synthesis, but we asked it to make a molecule with a specific color. So it went and it made a brand new molecule with a two-step reaction to get there, and it also plans the reaction conditions, it does the safety assessment, it plans out how to check the molecule, and we have this whole thing running from end to end, from intent, make a molecule with this, a chromophore with this wavelength, all the way to the end where it's made the molecule and it's got the UV-Vis mass spec readout. So of course, you have to evaluate this. Like I said, I want to leave some time for questions and answers. So we check these using human evaluations, or we go and execute the synthesis on the robot. So great. So basically, I just want to conclude here. So in a lot of tasks, having a low amount of data is a problem, but we can address this using things like Bayesian optimization and pre-trained models. Large language models can now leverage multiple tools. I showed you that, this ChemCrow thing, the way we teach it to use these tools, the way we taught it to use the robot, the way we taught it to use the literature search, is we just gave it like 2 or 3 sentences in English about how to use the tool and maybe an example. It's really remarkable, but that comes back to this idea that language has been designed to work in all these settings. Like, we write papers about how to synthesize molecules in English. So it's not surprising that it can, reason about how to synthesize a molecule in English. And so I think this is really the future right now, is these large language models being the glue to bring together all of the tools in biology and drug discovery. And of course, this is what we're going for. This is my vision of the future, and our vision of the future is that we take all of the tools that we've developed, turn them into English language, get all of our deep learning models able to express out in English language so that we can get these explanations in English and basically see what's possible now in this world of large language models. So with that, I'd be happy to take any questions and acknowledge my funding sources for this work. So thank you very much.